Welcome to the deep dive. Today we're jumping into some pretty advanced research, exploring the fundamental rules that govern biological structure.
We are. We're looking at everything from the tiniest molecules all the way up to the architecture of your entire genome.
And the focus is really on that link between form and function. We've got two technical reports that bring some surprising clarity to areas that have been, let's say, a little foggy for a while.
That's right. First, we'll look at how AI is being perfected to finally understand the really complex folding of RNA molecules.
Mm.
Then we're going to tackle a major scientific paradox. It involves the proteins that fold the genome, CTCF and cohesin, and it turns out they have this surprising dual life.
So our mission today is pretty straightforward. We're digging in to extract the unexpected insights.
Exactly. We want to understand how new computational power is revealing these hidden rules of molecular shape and how these tiny molecular switches are governing the most essential genes in your cells.
It's a journey from massive scale down to elegant, simple rules. So let's get into it. Okay, let's start small with the RNA molecule.
Right, ribonucleic acid.
It's this incredibly versatile molecule. It can carry genetic info, it can be an enzyme, it regulates all sorts of things. But there's a catch.
A huge catch. The function of any RNA molecule comes down to one thing: its specific 3D structure, how its long sequence folds up on itself.
And predicting that fold is notoriously difficult.
Oh, it's been a bottleneck for decades.
Yeah.
Scientists have used methods based on thermodynamics, calculating the lowest energy state, or they try to align sequences,
But those methods are flawed.
plagued by problems,
Mhm.
inaccurate parameters, or you just don't have enough similar sequences to compare it to.
So, of course, deep learning steps in. We've seen these powerful language models like BERT being repurposed for biology. They call them RLMs, RNA language models.
Yeah, but there's a problem with that approach, too. Most of these models, they just treat RNA as a one-dimensional string of letters, like text.
You're ignoring the physical reality, the fold.
Exactly. Or, even worse, they rely on those older imperfect tools to guess at the structure first, and then feed that flawed information into the model.
So the real challenge is, how do you teach a model that's designed for linear text to understand the spatial constraints of a real physical molecule that has to fold?
That's the question.
And what's the big leap forward here?
The solution is a model called RNA.
RNA. Okay.
It stands for Enhanced Representations with Base Pairing Restriction for RNA modeling. The team didn't just, throw more data at a standard model. They made a really clever design tweak.
And what was that?
They integrated a base pairing informed attention bias right into the pre-training phase.
Okay, wait, let me make sure I get this. This is a huge deal. So instead of feeding the model a complex, maybe even wrong, pre-calculated structure, they just gave the model the fundamental biological rule like A pairs with U, G pairs with C, and baked that right into how the model pays attention to the sequence.
You've got it. That's exactly it. They integrated domain-specific prior knowledge, which is really just the physics of base pairing, directly into the neural network.
So it biases the model to connect nucleotides that should pair up, no matter how far apart they are in that linear sequence.
Precisely. And this is where it gets really interesting. This simple, elegant tweak, it led to an emergent capability.
What do you mean by emergent?
Well, when they tested the model in what's called a zero-shot setting, so without any specific fine-tuning for predicting structure, RNA could do it anyway,
How?
just by analyzing its own attention maps. The model developed the ability all on its own.
I mean, think about that for a second. Zero shot means it wasn't even trained for that specific task.
Right. It was just trained on sequences with that one simple bias. But if you look at which parts of the sequence the model pays attention to when looking at another part, you can literally just read out the predicted structure.
It's predicting the fold organically from its language training.
Yes. And the data proves this isn't just a cool theoretical idea. In that zero-shot setting, its F1 score, a measure of accuracy, was 0.552.
And how does that compare to the old methods?
It blew them away. Established tools like RNA fold and RNA structure, which people have used for decades, they only scored around 0.49.
So the AI is better before it's even been specifically trained for the job.
That's the aha moment. And of course, once they did fine-tune it for specific tasks, the performance just soared. It's now state-of-the-art across a whole range of challenges.
Like what?
Well, on a big, complex data set called BPRNA 1 meter, its structure prediction F1 score hit 0.873. That's better than much, much larger models like you and I RNA.
So it's not about size, it's about the smart architecture.
It's about building in the right kind of knowledge from the start.
And it goes beyond just that 2D map, right? What about the full 3D shape?
Yep. It also achieves state-of-the-art for RNA contact map prediction, which is the crucial step towards that final 3D shape. It even handles really tough classification tasks and functional predictions like mean ribosomal loading.
MRL. That's a big one. That's a direct measure of how efficiently a protein is made from an RNA template.
It's huge. Because predicting MRL means we can assess how a change in RNA structure actually impacts protein output, which is fundamental to, well, almost everything in gene regulation.
So the big takeaway here isn't just that AI is getting better, it's a lesson in how to build these models.
I think so. Baking the fundamental physical rules, the base pairing constraints directly into the architecture is the ultimate shortcut. It's combining what we already know about physics with the raw power of deep learning.
That conceptual link, that fundamental rules dictate structure, is actually a perfect bridge to our second topic.
It is.
We're zooming out now from a single molecule to the 3D structure of the entire mammalian genome. And we're focusing on two proteins, cohesin and CTCF.
These two are universally seen as the major architects of the genome. They're responsible for folding about 6 feet of DNA into the tiny nucleus of every cell.
They create these functional compartments called TATADs and loops that bring distant regions of DNA together.
And the classic model, the one everyone learns, is that cohesin is the engine that physically pushes out these loops of DNA.
And CTCF is the stop sign. It's the anchor. It binds at specific spots, acts as an insulator, and makes sure the loops form in the right places.
Right. It's all about bringing a distant enhancer region close to a promoter to turn on a specific gene, critical for tissue-specific expression.
There's a paradox here, a big one that researchers have been wrestling with.
A very big one. If CTCF and cohesin are so essential, if they're the architects of everything, then getting rid of them should cause, well, chaos.
Right. You knock down the scaffolding, the whole building should collapse.
Exactly. The transcription of thousands of genes should go haywire.
But when researchers acutely depleted cohesin, what did they find?
They found minimal chaos. The loops themselves, the physical structures, they were globally disrupted. The scaffolding definitely came down.
Okay.
But the resulting changes in gene transcription were shockingly limited. For example, getting rid of cohesin only downregulated about 2% of all expressed genes.
Wait, only 2%. If these proteins are the undisputed architects, that sounds like the blueprint barely matters.
It's a huge contradiction.
I mean, that completely challenges the prevailing view that this 3D looping is the main way genes are regulated.
It creates a major conflict. If their architectural role only impacts a tiny fraction of genes, they must be doing something else.
A non-architectural job.
A massive non-architectural job. They must be acting as direct transcriptional regulators, completely independent of those big loops.
So the research shifted. The new goal was to find this hidden function, to figure out what specific genes are governed by this secret role.
Yes, and the key was to separate the two functions. They found a subset of genes that were shut down when CTCF was removed,
Okay.
but were not affected when they used a chemical that specifically blocks the activity of those long-range enhancers.
So they isolated the genes that CTCF controls without using loops?
Precisely. And what were these genes?
Let me guess, not the specialized tissue-specific ones.
Not at all. They turned out to be broadly expressed housekeeping genes.
Wow. The essential maintenance machinery.
Exactly. Genes like NLE1, BISL, POL3A, these are what the DepMap project calls common essential genes, the ones every cell needs just to survive.
This changes everything. So the core reason CTCF is essential for a mammalian cell to live isn't its fancy architectural job.
It's because it directly activates the basic non-negotiable genes required for life.
That is profound. Its most fundamental job isn't building the beautiful 3D structure, it's just keeping the lights on.
Yes.
Okay, so if it's acting as this direct switch, what are the rules? How does it work?
It is incredibly precise. Just like the base pairing rules in RNA, CTCF's non-architectural function is dictated entirely by its position and its polarity, the orientation of its DNA binding motif relative to a gene's start site.
So where it binds and which way it's facing determines what it does.
Completely. Think of it as having 3 different identities.
Okay, walk us through them.
Identity 1 is the activator. This happens when its motif is forward-oriented and it binds just upstream of the gene's start site. It turns the gene on.
Simple enough. What about the repressor rules?
Identity 2 is the first type of repressor. It binds a bit further upstream, and its job is to block antisense transcription, which is like noisy interference that can mess with the main gene signal.
And identity 3.
That's the second repressor function. When CTCF binds right inside the core promoter, or just downstream of it, it physically blocks the main reading of the gene.
So you have one molecule that can be an on switch or two different kinds of off switches, all depending on its parking spot.
Exactly.
That kind of complexity makes you wonder, are these two huge jobs, the architect and the direct regulator, handled by different parts of the protein itself?
They are. The research dissected this beautifully. They found the architectural function, the part that anchors cohesin and deals with cell-type specific genes, is all handled by the N-terminus of the protein.
The front end.
Right. And the non-architectural functions, specifically activating those essential housekeeping genes, are mediated by the C-terminal domain, the back end.
And that physical separation must explain why it's so essential for our cells.
It does. It seems to be that C-terminal activator function that is universally required for proliferation in mammalian cells.
Which brings us to CTCF's cousin, Boris. This is the crucial comparison, right, for understanding the evolution of all this.
Absolutely. Boris shares the same DNA binding part as CTCF, the zinc fingers, so it sticks to the same DNA sequences. But critically, Boris cannot anchor cohesin loops.
It lacks the architectural skill.
It does. But when scientists reanalyzed old data, they found that Boris is a competent transcription activator.
So it can turn on genes.
Yes, it can actually restore the expression of those same enhancer-independent CTCF targets, the housekeeping genes.
So Boris can't build the mansion, but it can make sure the furnace stays lit.
Precisely. And this functional divergence helps explain why different species have different dependencies on CTCF.
How so?
Well, mammalian CTCF is essential because of that C-terminal activator role. But in simpler organisms like Drosophila fruit flies, their CTCF primarily just acts as an insulator. It doesn't anchor loops, and it's actually dispensable for the fly's cells to proliferate.
So the evolutionary step that made CTCF essential for complex life like ours wasn't about perfecting its role as an architect.
It seems not. It was more about gaining that critical C-terminal activator function to make sure the basic survival genes were always reliably switched on. That seems to be the really definitive story of why it's so essential.
This deep dive has spanned massive scales, but the insight that keeps coming back is surprisingly consistent. Structure dictates function.
And that structure is always determined by a foundational set of rules.
At the molecular level, with RNA, we saw the model use the basic rule of base pairing.
And integrate that knowledge right into its attention mechanism to gain this incredible emergent ability to literally see RNA structure.
And then at the cellular level, we found the genome's chief architect, CTCF, whose most critical function depends not on the gene it's near, but on its polarity and its precise position.
Acting as a high-precision, location-specific switch for the most important genes we have.
In both of these stories, the key to prediction, the key to understanding function, is identifying which pieces of prior knowledge are the most important.
Is it the AUGC base pairing rule? Or is it the precise orientation and position of a binding motif? Finding that influential piece of information is everything.
So understanding the rules of structure is really the ultimate shortcut to predicting function and identifying the essential mechanisms that maintain life.
And that leaves us with a provocative final thought for you. Given that these absolutely essential functions, like turning on your core housekeeping genes, seem to be divorced from the complex, highly regulated architectural functions of the 3D genome,
Yeah.
does this suggest that the most basic rules of life are inherently simpler and maybe more robust than the mechanisms we've built on top of them for complexity and specialized development?
Welcome to the debate. Our focus today is on the central infrastructure of the genome, the molecular machinery responsible for organizing our DNA in 3 dimensions. We're really diving into recent findings regarding the CTCF protein, that's the CCCTC binding factor, and its critical architectural partner cohesin, primarily RAD21. These proteins are, well, they're the keystones of genome folding and gene expression in vertebrates.
Right, and the data detailing CTCS function has truly exploded in complexity. I mean, we can no longer view it simply as some kind of static glue. So the central question before us is whether CTCF's primary indispensable role remains structural and architectural, meaning, insulation and anchoring loops, or if its newly characterized non-architectural roles, like direct transcriptional activation and repression, represent a distinct and functionally essential mechanism that explains why it is so indispensable in mammalian cells.
And I'm going to hold that the architectural role, which is the mechanism mediating essential enhancer-promoter contact through loop extrusion dynamics, remains the primary and, frankly, most consequential function, especially when we consider cell type-specific regulation.
And I'll contend that the non-architectural role, specifically its ability to directly activate core essential housekeeping genes, is a functionally divergent and critical mechanism. I think this specialized role is what truly establishes mammalian CTCF's indispensability, and it fundamentally challenges the completeness of a purely architectural model.
Okay, so the foundational role of CTCF and cohesin is fundamentally structural. The folding of the genome into loops and topologically associating domains, or TADs. This structural order is what physically promotes EP interactions, ensuring cell type-specific enhancers activate only their correct targets and, well, maintaining genomic stability by preventing misactivation. We have just overwhelming empirical proof linking cohesin activities specifically to this enhancer-dependent regulation.
What is the most definitive evidence supporting that architectural bridge requirement?
Well, the evidence surrounding the CBP300 acetyltransferase inhibitor A485 is just profound. CBP300 activity is the hallmark of active engaged enhancers. When we look at genes downregulated by acute depletion of cohesin, specifically the RAD21-AID complex, those genes are overwhelmingly dependent on active enhancers. We observed that 86% of the genes downregulated by RAD21 loss also overlap with genes also downregulated by A485 inhibition.
86%.
Right. And to put that statistical overlap into a biological context, the resulting odds ratio is 80.
80 is certainly a high number.
It is. It's a dependency that is nearly absolute. It shows that when cohesin is required for a gene's expression, the activity of the enhancer, the CBP300 modification, is virtually guaranteed to be necessary as well. So this confirms that the structural bridge provided by cohesin is the operational mechanism for enhancer communication. Moreover, we know this architectural function maps directly to the CTCF N-terminus, which is required for cohesin retention. The major gene regulation effects we see upon loss of RAD21 or CTCF stem directly from disrupting this foundational structural architecture.
Okay, I see that, but I come at it from a different way. Because the purely architectural view, while it's incredibly elegant in its simplicity, it fails to account for several key functional discrepancies. I mean, if CTCF's sole vital role is to anchor loops for cohesin, why does the acute depletion of CTCF or cohesin only regulate a relatively small fraction of the genome? And more puzzlingly, why do the genes regulated by RAD21 and CTCF show such a remarkably limited overlap? Their transcriptional changes only correlate with a low Pearson correlation coefficient of 0.18. That's a profound lack of coordination between two supposed partners.
A difference in timing or stability could explain that correlation deficit, not necessarily a completely different function.
I disagree. I think that low correlation suggests functional divergence that is mediated by a distinct non-architectural role. CTCF acting as a position-specific transcription activator or repressor, promoting or hindering poly-T activity directly at the promoter. And this role is structurally mediated by the C-terminal domain residues 578 through 736. Crucially, this C-terminal function is essential for cell proliferation, and it regulates a distinct, non-enhancer-dependent set of genes. You're referring to the housekeeping genes? Yes. These are broadly expressed, common essential genes like NYWEL1, BiSil, and POLR3A that are downregulated by CTCF depletion but are critically not affected by the A485 inhibitor. That not affected part is the linchpin. It tells us their regulation is entirely enhancer-independent, fundamentally separating them from the architectural mechanism. This C-terminal dependent, enhancer-independent activation is also the evolutionary distinction. Mammalian CTCF is indispensable for cell proliferation, whereas Drosophila CTCF, which lacks this specific C-terminal transcriptional activation function, is dispensable. The necessity lies outside the loops.
Okay, let's unpack that supposed divergence. I agree the correlation, the PCC, of 0.18 is modest, but this doesn't invalidate the architectural model. Let's look at the intersection, not the divergence. If we analyze the targets that are downregulated by both acute RAD21 depletion and acute CTCF depletion, the shared targets, the architectural role becomes, I think, undeniably primary.
But why is the convergent set more important than the divergent set?
Because the shared targets represent where the structural requirement is absolute. And what do we see in that converged set? They are overwhelmingly CBP 300 dependent, showing a 93% overlap with A485 downregulated genes, again with an odds ratio of 71. This is the crucial finding. When we simultaneously remove the loop extrusion engine RAD21 and the loop anchoring foundation CTCF, the common resulting effect is the complete collapse of enhancer-mediated regulation. This convergence proves that the most biologically consequential function shared by both proteins is the structural one.
That's an interesting point, though I would frame it differently. While the shared set demonstrates that CTCF is required for cohesin's architectural work, the low correlation between the full regulated sets proves that CTCF performs essential functions beyond simply anchoring the complex. You're focusing on the convergent set, which is small. I look at the divergent set because that's where the mechanism of essentiality lies.
Which is the CTCF down A485 indie set.
Precisely. We call them the CTFC-dependent genes that are non-dependent on enhancer activity, A485ND. These genes are broadly expressed across tissues and activated by CTFC independently of the looping mechanism. If the only truly vital role of CTFC was architectural, we would expect a near-total transcriptional convergence between RAD21 and CTFC loss, particularly in the most essential gene sets. The observed divergence, coupled with the evolutionary dispensability of the Drosophila counterpart, proves the necessity of this C-terminus-mediated transcriptional activation mechanism. This brings us squarely to the separation of domains. We've demonstrated that the essentiality of CTFC and cell viability stems from activating those housekeeping genes, a function mediated by the C-terminal domain. We found that loss of the N-terminus, which handles cohesin anchoring, impairs proliferation less severely than loss of the C-terminus.
This functional separation between the loop anchoring N-terminus and the indispensable regulatory C-terminus argues forcefully for true functional dualism, mechanistically and genetically separable roles.
I see why you think that, focusing on that structural separation, but I would argue that the C-terminus may simply be the execution module for a position-dependent architectural outcome rather than a completely divorced independent function. For example, consider the CTCF paralog Boris or CTCFL. Boris shares similar DNA binding capability, but critically lacks the ability to anchor cohesion.
Right. And Boris can restore the expression of those housekeeping genes, the enhancer-independent targets. That is strong evidence that a non-architectural direct activation function exists.
It is, but here is the critical distinction. Boris cannot restore the expression of the enhancer-dependent cell-type-specific targets. This failure demonstrates that the N-terminus's ability to anchor cohesin loops, the architectural function, is the defining mechanism for the broader dynamic cell-type-specific regulatory program. The C-terminus supports the housekeeping genes because those genes require robust, localized constitutive activation, often lacking the extensive long-range EP context typical of cell-type-specific genes. The C-terminal function is localized support, but the N-terminal architectural function is the master regulator of the entire dynamic genomic structure.
That attempts to force a transcriptional role back under the umbrella of architecture. But the mechanistic specificity of the C-terminal action points to a dedicated transcriptional factor role that is fundamentally separate from coarse loop dynamics. Let's look at the third point: how fine-grained this non-architectural control is at the promoter itself.
Well, the architectural model already acknowledges that CTCF binding serves as an enhancer blocker and facilitates EP interaction by halting loop extrusion. We know the function is highly dependent on its binding polarity, which dictates chromatin looping direction.
Polarity and position matter profoundly, but the functional outcomes we observe are too nuanced,
to be explained solely by loop physics. We find that CTCF can function as both a direct activator and repressor of promoters, with the outcome dictated by exact position and orientation relative to the transcription start site, or TSS. Stream of the TSS, at a median of around 79 base pairs, results in transcriptional activation. This is the core housekeeping activation mechanism. However, when CTCF binds further upstream, it completely represses upstream antisense transcription, USTRX, acting as a roadblock to transcription coming in the opposite direction.
And you also find that binding within the core promoter region or just downstream of the TSS results in repression of sense transcription?
Exactly. We have a protein that can achieve 3 fundamentally different regulatory outcomes: activation, upstream repression, and downstream repression, all based on its specific motif orientation and its location within a window of just a few hundred base pairs. This level of nuanced, position-dependent regulation, which affects Pol2 recruitment and elongation directly, screams of a dedicated transcriptional factor role. It cannot be fully reduced to the relatively coarse, long-range mechanism of loop extrusion and halting. It's a localized, highly specific regulatory dial that is absolutely essential for cell proliferation, as shown by the unique essentiality of mammalian CTCF compared to its dispensable fly counterpart.
While I concede the high level of specificity for those local, non-architectural functions at the promoter, the overall data confirms that the structural component, the N-terminal dependent interaction with cohesin, and the resulting long-range EP contacts dictates the fate of the vast majority of cell-type-specific and developmental genes. This is the organizational mechanism responsible for generating genomic complexity and driving differentiation across the entire organism.
But essentiality, which is the core of our debate, isn't driven by differentiation. It's driven by conserved core metabolic requirements. The evolutionary acquisition of this function, the structural separation of domains, and the isolation of the enhancer-independent housekeeping gene set, the CTCF-down A485ND genes, all point toward a required functional dualism. The C-terminal domain provides the critical enhancer-independent activation necessary for conserved cell viability, a role the N-terminal architectural domain, powerful as it is, cannot fulfill alone.
Our discussion has, I think, effectively highlighted the dual nature of CTCF. However, I maintain that its architectural role, demonstrated by the strong quantitative link between RID21-regulated genes and CBP300 targets, fundamentally controls the expansive, dynamic, cell-type-specific regulatory landscape. While the non-architectural function provides localized support for housekeeping genes, the N-terminal dependent loop anchoring mechanism remains the most consequential for dynamically regulating gene expression through long-range EP interaction.
This has certainly clarified that the architectural function is crucial for enhancer-dependent genes and the complexity of cell-type regulation. Nevertheless, the compelling evidence for the C-terminal dependent activation of conserved, essential housekeeping genes, coupled with the fine-grained position and orientation-specific repressive and activating roles near the TSS, forces us to adopt a model of true functional dualism. CTCF is essential not merely because it organizes the genome architecturally, but because it also functions as a sophisticated, position-specific transcriptional factor, a distinction that's vital for understanding its full, indispensable biological role in mammals.
The material clearly shows how cohesin and CTCF integrate both these powerful functional modes, regulating distinct and overlapping gene sets through enhancer-dependent and enhancer-independent mechanisms. Understanding the precise coordination and really the evolutionary balance between these two functional classes is where the future of this research lies.